hypothalamus and FSH, all right, we're going to have the same sequence. So the hypothalamus nuclei are going to release GnRH. And GnRH reaches the anterior pituitary.
Is this the making of inhibition?
And then we're getting to that, along with another molecule. And then the anterior pituitary same cells that make LH are going to synthesize FSH, okay? Which stands for follicle stimulating hormone. Both the words LH, luteinizing hormone, and follicle stimulating hormone refer to the actions of these molecules in the female. So oftentimes they're just used interchangeably. Sometimes they're given different names. So LH is also known as interstitial cell stimulating hormone. Okay, so when you read that, it's just referring to its role in the male. So FSH now is going to travel via the blood, and it binds G protein coupled receptors on subtentacular or Sertoli cells. Sertoli cells are shorter to right. So testosterone passes through the plasma membrane of the Sertoli cells. FSH is binding to a membrane receptor. So down here on the accessible level to the bloodstream, we're going to have FSH binding to its membrane hormone. And just like LH, it goes through the adenylyl cyclase pathway and will induce increased levels of cyclic AMP within the subtentacular Sertoli cell. Now, there are a variety of functions, but as far as hormones or contributing components, two molecules will be synthesized as a result of binding FSH. One is the inhibin that we discussed earlier. So inhibin binds to anterior pituitary cells and decreases LH and FSH secretion. The other molecule that is made is not a hormone in the sense that it gets to the blood and travels all over the body, but functions specifically at the subtentacular Sertoli cell component. So it functions more like an autocrine, because it works on the cell that makes it. So this is known as androgen binding protein. It binds, remember what I said androgen was? Forms of testosterone. So androgen binding protein binds testosterone, and it keeps 90% of the testosterone made by the Leydig cells in the subtentacular cells. So 10%, the rest is circulating throughout the body and having systemic effects. But 90% of the testosterone is maintained within the subtentacular cell by binding onto androgen binding protein. So this functions maintain a high concentration of testosterone. Yes?
So what happens if the androgen receptors start degrading?
Being broken down?
Yeah.
There's a complex relationship if you have high levels of testosterone. There's actually a decrease in the amount of receptors that are made. So if the testosterone is broken down or degraded, then a couple of things you'll see that. Well, hold on that thought for a minute. I'm going to go through and talk about what it does do. Okay. One of the components of the basement membrane associated with the sustentacular cell is to maintain what's known as a, remember the blood-brain barrier we talked about with the tight junctions protecting the brain? Well, there is a blood-testis barrier. So that the spermatogonia are outside this blood-testis barrier. Once, however, they start to be processed in forming the primary spermatocytes, the testosterone helps these primary spermatocytes to get through that blood-testis barrier, and then afterwards maintain the tight junctions that prevent exposure of these developing spermatocytes to anything that's in the blood or connective tissue. So it both helps to maintain the blood-testis barrier and then to help isolate, to help the cells get through and then to help maintain it and isolate the cells out there. Yes?
You're saying testosterone does that?
Uh-huh. And then testosterone also, interestingly enough, remember how I talked about the fact that as these cells are migrating through, there are molecular connections between the developing spermatocytes and the plasma membrane of the sustentacular cell. And testosterone helps to maintain that as well. So when testosterone is low, too low, then these cells do not further migrate. They stop their development. Interestingly enough, the opposite occurs, and it depends on its location in the cell. Testosterone also helps separate those junctions, break down those junctions, so that when we have our late spermatids developing out here, they are free to become in the lumen. With very low levels of testosterone, that separation doesn't occur, and those late spermatids that are almost mature spermatozoa are degraded and broken down and metabolized by the Sertoli cells. So testosterone is involved with the Sertoli cell in the cell division and movement and changes that occur to the sperm as they go through, and their ability to actually get out of, happen. They separate from there. Now, so there's a lot of argument sometimes about which direct functions sustentacular cells have versus what the testosterone component is. Sertoli cells provide this environment. They provide the nutrients to the developing spermatocytes. So leaving this plasma membrane and going into the Sertoli cells are all of the new amino acids and new lipids that they need as they're going through meiosis. And then when the early spermatid changes to the late spermatid and we get rid of extra cytoplasm, the Sertoli cell degrades that. So it kind of is digesting that. If the late spermatid is not able to leave, it eats the whole cell. Right? So its normal role is to eat the extra cytoplasm removed from the early spermatid as it changes from a round cell to a long, elongated, basically nucleus. But if the late spermatid doesn't separate, it just eats the whole thing. You can see there's a lot of steps in here where that is regulated. The sesentaculated cell also produces the fluid that helps to propel the sperm and push it along and out of the lumen of the seminiferous tubule. So again, back to, we've listed several different chemicals here. So all of this stuff right here in the laid egg, from cholesterol to testosterone, is LH regulated. So it helps you remember that. LH actually stands for luteinizing, but if it helps you remember the L for Leydig cells, you can connect it that way. The end result is testosterone. Most of the testosterone ends up being associated with Sertoli cell, not in the developing spermatocytes, but in the Sertoli cell, regulating its activities. Then the Sertoli cell binds FSH and makes these molecules. Both LH and FSH working via cyclic AMP components. All right, now let's look at spermatogenesis. So this is